Hello, welcome to D-board exams. This is a biochemistry practice. Most of you have requested certain topics, which includes TMJ, also includes biochemistry, and there are other topics such as microbiology and physiology. If you do request a certain topic, we can always go through that. Okay, as I mentioned, usually I guide students, okay, through both national board part one and part two. Make sure they focus on certain areas which is going to maximize their score. And mostly, I give usually online practice exams, okay, which are timed with a full explanation at the end. So some of these discussion here. It's questions with discussion, mostly of the topics which, if you're going to take the dental boards, you have to be aware of these topics and make sure you understand them or at least memorize them. Okay, so that you know them really well. So here I have a structure of glucose. Okay, so this is a glucose molecule in a fissure. So this is a fissure configuration. Now, why did I put glucose here? Because glucose is essential, especially if you're studying for this exam. You need glucose so you can break it down to ATP. Okay, and mostly your brain uses up glucose. Okay, that's the most energy source of the brain is glucose. So make sure if you're studying and for good memory, make sure that you maintain your glucose levels. Okay. All right. So let's go to the first slide. So the first slide here, the topic is collagen. Some of these questions, some of the previous released questions, but we'll go ahead and go through them and discuss, okay, so that you know exactly what you need to focus on. So the question on collagen: which of the following collagen synthesis, okay, post-translational? So you have to pay attention. It's usually post transcription. Okay, transcription is making of the RNA. Translation is making of the protein itself. Okay, so which of the following collagen synthesis post-translational events occurs extracellularly? Okay, is it collagen? Some of the amino acids in collagen undergo both hydroxylation, okay, and also glycosylation. So is it does glycosylation occur outside the cell? Does hydroxylation occur outside the cell? Does intramolecular cross-linking of collagen peptides occur outside the cell, or is formation of disulfide bonds form outside the cell? Okay, so here you have to know collagen synthesis. Okay, make sure you know the types of collagen. Make sure you know that collagen is a propeptide. Okay, mostly when it's released from the enzymes, it's released as a pro collagen. Okay, so that's how it's released. Now the answer here, just so that I give you the answer. So the answer here is going to be intermolecular cross-linking. Okay, cross-linking. So usually the collagen peptides are formed, and then three of them are cross-linked together to form the collagen fiber. Okay, so And this process happens outside the cell. The synthesis of collagen. So for example, if you have a cell here, okay, so you have your endoplasmic reticulum, okay. Remember, ribosomes, and this is your cytoplasm. I am a biochemistry major right now. I've graduated. I've been practicing for a while now, but I love biochemistry. Okay, so cytoplasm, take this as ER. Okay, endoplasmic reticulum is ER. Now remember, the endoplasmic reticulum, that's where the ribosomes. Okay, let's do this as a ribosome. That's where ribosomes attach. Okay, when they're about to make protein. Okay, so the ribosomes, when they're about to make protein, they attach onto the endoplasmic reticulum. Now, you have to know the function of endoplasmic reticulum. Remember that endoplasmic reticulum, it's like a transport system. Okay, so the ribosome attaches to the endoplasmic reticulum so that when they make the protein, then it can be transported by the endoplasmic reticulum to other areas of the cell. Okay, for example, the cell membrane. Now you have collagen being produced here. Okay, so this ribosome translating and forming the collagen inside. Now this collagen ends up being transported, okay, to outside the cell. Okay, so it's released to outside the cell. So now this is extracellular. Okay, and outside the cell, the collagen is released as procollagen. Okay, and then this procollagen, you have to have 3 of them. Okay, now this collagen, remember, you have to know exactly what forms up collagen. It has a lot of glycine. Okay, so it has a lot of glycine, but it also has, so collagen has glycine, it has proline, and it has lysine. Okay, so most of these molecules are abundant, and the most abundant in collagen is glycine. Okay, so when you think about collagen, think about glycine. It's the most abundant amino acid in the collagen. However, the proteins which undergo hydroxylation and glycosylation, okay, are proline and lysine. Okay, so proline and lysine, they undergo both hydroxylation, okay, and glycosylation. And this glycosylation, so this is the A and B. Okay, these are the process, and these occur inside the cell usually. So when they occur inside the cells, for the hydroxylation to occur, you need vitamin C. Remember, when you see collagen, think about, okay, collagen, think about vitamin C. What's the other name for vitamin C? It's ascorbic acid. Okay, all right, so ascorbic acid is the cofactor. You need vitamin C for the collagen to form and become strong. Okay, to cross link, and that cross linking happens because you have glycosylation, hydroxylation of of proline and lysine. All right. So this presentation mostly is focusing you on the things that you have to know before you go and sit for your exam. All right. So our answer here is C. Okay, as I mentioned. Okay. So make sure you review this, and make sure you memorize it and know it. Okay. So the next topic is cytochrome. Okay. So cytochrome. The question is, which of the following is an essential element found in cytochrome? Okay. So for you to answer, you have to know what cytochrome is. Okay. So cytochrome, these are it's a quaternary. Okay. It's a quaternary structure. Quaternary protein. Okay. And this quaternary protein actually has, just remember that, like hemoglobin, hemoglobin, just like hemoglobin, which is a quaternary structure, also has heme, okay? Heme as cofactor. So cytochrome, think about when you think cytochrome, okay? Think about heme. Heme, okay? And I'll tell you why you have to know. When you think about cytochrome, think about heme. Okay. Now, heme, as you know, when somebody is anemic, okay, you may say, "Oh, have you been taking your iron?" Okay. So iron is usually also a cofactor in the heme. Okay, it's always found on heme molecules. Okay. This always helps in oxygen carrying capacity. Now, in cytochrome, the heme has iron. Okay, so our answer, I've already mentioned the answer. So our answer is going to be C here. Okay, as iron. And yeah, so C as the answer. Now, the cytochrome itself, since it has iron, it uses the iron to transport electrons. Okay, it uses iron to transport electrons, and this is important because you have to know, we're going to discuss about mitochondria. I love mitochondria. Okay, the electron transport chain. So you have to understand all this, that this is essential in transporting electrons, so you can generate ATP. Okay, ATP is the most important one of the most. It's a energy currency of the body. Okay, that's why I put a glucose molecule at first, because the glucose molecule is broken down to ATP via glycolysis, and also via oxidative phosphorylation. You have to know glycolysis. You have to know oxidative phosphorylation. You have to know the structure of the mitochondria. Okay, mitochondria has two membranes. There's an outer membrane, and also has an inner membrane. So you have to know where the oxidative phosphorylation occurs, and you also have to know where the glycolysis occurs. Okay, so just to say, when you see cytochrome, okay, think about heme. And then if you see him, think about iron. Okay, iron. All right. So our next one is elastin. Okay, elastin. Usually I say elastic because that's the property of it. Okay, so elastin gives elasticity. Gives elasticity to organs such as skin. Okay, and collagen gives support. For example, tendons. Okay, they keep the organs in the right place. Okay. For example, they prevent the shoulders and the ankles from dislocating. Okay. They protect. Okay. Collagen protects organs such as the heart. You know, the heart is surrounded by pericardium. Okay. So you need collagen, and you need elastin. Okay. Now, they both to prevent the one structure from becoming too rigid, or the other one from becoming too elastic. You have to the organs usually have both elastin and collagen. Okay, but you have to know the comparison between the two. Make sure that before you go to the exam, you have to know the comparison. So, which of the following do elastin and collagen have in common? So this is a compare and contrast. Okay, so easily stretched. Okay, easily stretched collagen, no. So this is only elastin. Okay, so this is elastin. Okay, so A is only elastin. B, absence of proline. No, they both the absence of proline. So absence of proline mostly is not common. Okay, both of them have proline. Okay, and so the absence here is makes it wrong. There is sulfide cross-linking. Not necessarily. Mostly it's glycosylation. Okay, and the hydroxylation. That's what causes cross-linking. A triple helix. So this is the property of collagen, and it's the elastin does not have a triple helix. So as I told you, glycogen is made up of glycine mostly, and also elastin it's like 1/3 is elastin. So this is the answer. Okay. So that's so our answer here is E. Okay. So you have to know you have to know compare and contrast collagen versus elastin. Okay. The other common thing, they are both released as propeptide, meaning proelastin or tropoelastin, and then the other one is released as a procollagen. Okay. So collagen may have usually it has a different gene. You have type 1, type 2, type 3, but however elastin is only transcribed by or encoded by one gene. Okay. Also, you have repeating units of collagen. Collagen, you have repeating units of glycine. Okay, which is, and then you have X and Y. So where the X is proline and the Y can be lysine. Okay. All right. So usually in the elastin, so elastin, both elastin and collagen, you have proline. Okay. However, in elastin, you don't have hydroxylysine. Okay, that's missing. So you have to know. So this is just a quick review of what you need to concentrate on. Okay. So collagen and elastin comparisons. Next topic. Vitamin K. All right, vitamin K. You have to know vitamin K. Don't go in the exam without knowing vitamin K. Vitamin K is very important, especially on patients taking certain blood thinners. Okay. Such as coumadin. Okay. You have to know vitamin K dependent coagulation factors. Some people say 1972, like 2, 7, 9, and 10. Okay, that's how they remember. Some people remember it. That's how I tell my students to memorize it. However, there's so many ways to memorize. I've never used 1972 because I'm more of a cascade. So I usually memorize the coagulation cascade. So that's that was because, of course, I was a biochemistry major. That's the reason. So a question is, a derivative of vitamin K is a coenzyme for which of the following? All right. So you have to know, before you look at this question, you have to know what vitamin K does. Okay. So vitamin K helps in when I mention this, you already know the answer. It helps in an enzyme. Okay. It's a epoxide reductase to do carboxylation. So to facilitate carboxylation of glutamate side chains in the coagulation factors, and that also increases calcium binding to the amino to the coagulation factors, which helps with the blood clotting. All right. So carboxylation, make sure you know that. Okay. Carboxylation, make sure you know the carboxylation. Okay. And of the glutamate, make sure you know those. Okay. And the other thing you have to know the enzyme which helps which needs vitamin K to conduct carboxylation of glutamic acid. Okay, so the answer here, as I mentioned, is E. All right, well, make sure you review and know these topics. All right, one of my favorites, oxidative phosphorylation. Now, when you hear, for somebody who has not taken biochemistry before, or just took biochemistry briefly and did not go into details, when you see the term oxidative phosphorylation, okay, don't be scared. They say, oh, what does this mean? Okay, remember, ATP, okay, is needed. Oxidative phosphorylation, another word is called respiration. In other words, okay, it's respiration. So it's respiration. Remember, for you to respire, when you are respiring, you need oxygen. Hence, the oxidative. Okay, so you need oxygen. So, and you need oxygen to make ATP. ATP stands for adenosine. All right, triphosphate. Okay, now you have a phosphate. So oxidative, you need oxygen. Phosphorylation, you need to add phosphorus. Okay. Now, what are you adding phosphorus to? Is you have ADP. Okay, so you need oxygen and you need phosphorus or phosphate, okay, to make ATP. Hence the name oxygen phosphorus. So oxidative phosphorylation, which is the same as the respiration for you to make energy. Okay, so do not be confused when you see this term and say, oh, oxidative phosphorylation, what is it? Okay, now oxidative phosphorylation, you need energy. Okay, you need energy generated from the electron transport chain in the mitochondria to generate ATP. Okay, so it's oxidative. and you also need oxygen. All right. Now, remember, there's another source of there's another what we call glycolysis. Okay. In glycolysis, so the difference is oxidative phosphorylation happens inside the mitochondria. Glycolysis happens in cytoplasm. Make sure you know that. Okay, those are the differences. Glycolysis produces less ATP, less energy compared to oxidative phosphorylation. Make sure you know how much ATP is produced by glycolysis. Make sure you know how much ATP is produced by oxidative phosphorylation. So, what's the question here? The question is, during oxidative phosphorylation, the energetically unfavorable proton gradient, okay? The energetically unfavorable proton gradient is created using energy from what? Okay? So you have to know how the energy is generated. So mostly, I'll draw I'll draw the mitochondria here. Just a quick rendering or figure or depiction of mitochondria. Now remember, mitochondria is found inside the cell. For example, this is a cell. A cell has a cell membrane. Now mitochondria also has cell membrane. Now let's say that this is a mitochondria which is big. Okay, it's not going to be as this big. Okay, so this is mitochondria. Now this is the outside membrane, and then you have an inner membrane. Okay, so you have an inner membrane. So this is a cell wall, and then you have this here is the mitochondria. Okay, outer membrane, and then inside you have another membrane. So this is inner membrane, and then you have like complexes. You have like enzymes. Okay, so you have enzyme 1 here, you have like enzyme 2 here. Okay, you have enzyme 3, sometimes just go straight to 4, and then you have 4 and 5. Okay, so usually 5 in humans, 5 the complex 5 is the one which is ATP synthase. Okay, so remember, you have here, so this is inner outer membrane, you have inner membrane, so there's a space here in between. There's a space. That's what we call intermembranous space. Okay, in the mitochondria. So the NADH, okay, remember the citric acid cycle, which you also have to know. The other name of citric acid cycle is Krebs cycle of or citric acid cycle. Okay, so you remember the Krebs cycle produces NADH and also citric the succinate. Okay, so succinate and NADH are produced. Those, they have electrons. So those electrons are carried to these complexes here. Okay, and then those complexes, once those electrons are accepted, then they produce a proton. One of the proton is pumped outside into the inner mitochondria, so the intermembranous space in the mitochondria. So you have protons here. Okay, so these keep pumping and in here in this space you have a lot of protons. Okay, I'll put protons. Okay, so these protons, as electrons are being transported, okay, across each of the complex, these red dots here, then you pump the protons into the space between the two membranes. All right, and then at the end, okay, usually there's a question which asks which molecule receives the electron. Usually the ATP synthase, okay, so electrons are transferred. The electron acceptor in respiration or oxidative phosphorylation is oxygen, which is the reason we need oxygen. Okay, so the oxygen takes up those electrons, and then one of the hydrogens, the protons, combines with that oxygen with the extra electron and forms water. So that's why you have the end product of respiration. You have ATP, which is the energy currency, and water. Okay, that's the reason you need oxygen. You need oxygen because it's the electron acceptor. All right. So this energy to create ATP comes from the electron transfer. So as electrons are transferred across the membrane, you get ATP. Okay. So our answer. So our answer here is D. Okay. Let's see. Okay, so let me see. Trying to bring up the answer here. Okay, sorry about that. Okay. Yeah, there it comes. So that's our answer is D. And our next topic. Okay. Let's go to our next topic, which is let's see. Oh, this is going back here, so let's go forward. Okay. Our next topic, which I think you should know before you go into your exam, is cholesterol. Okay. Cholesterol, you know, the number one killer in America is heart disease. Okay. Heart disease is associated mostly with high cholesterol. Okay. Atherosclerosis because of high lipid diet. Okay. So you have to know, and you know the medications for treating high cholesterol. You have to know all those things. So the question here is, which of the following is necessary for de novo, like new synthesis, okay, of cholesterol? So here, the cholesterol, okay, you have to know that what you have to really know the biochemical pathway, okay? You have to know the biochemical pathway of how cholesterol is synthesized, okay? So there's so many steps, but remember that cholesterol is synthesized from the breakdown of fatty acids or acetyl-CoA, okay? Acetyl-CoA, which is also acetyl-CoA is also the end product of glycolysis. Okay, you have to remember that. So acetyl-CoA or acetyl-CoA, okay, and then you have you can this conversion here into cholesterol. You need, okay, to convert this into HMG CoA, you need the enzyme, which is called, of course, there's almost the same name, HMG CoA. That's a synthase. Okay, so you're making HMG CoA. Now, this enzyme, HMG CoA synthase, okay, forms HMG CoA. Now, HMG CoA is going to be converted to mevalonate acid. Okay, so this mevalonate mevalonic acid or mevalonate, this here, okay, for you to move from HMG CoA to mevalonic acid, you need ATP. I mean, sorry, not ATP, but NADPH. So you already know the answer. Now you know that the answer is D. So you need NADPH, and not only do you need NADPH, but this NADPH is needed by the enzyme HMG CoA reductase. You are reducing it, HMG CoA. Okay, so when you reduce HMG CoA, you make mevalonate, okay, which is a precursor to forming the cholesterol. Now, mevalonate is further converted, okay, after multiple steps. So there's a lot of steps here. It's converted to scalene. Okay, make sure you know scalene. Okay, that's the reason I'm writing it, because it's important key. And then the scalene is further converted, okay, after multiple steps, okay, to cholesterol. So make sure you look at the biochemical pathway, okay, of how cholesterol is formed. But you see at this step here, you need NADPH. Now, don't be confused. It's not NADH, okay? It's NADPH. So NADPH is needed by the enzyme, the HMG CoA reductase. Okay. Now I was about to I slowed down because I was thinking about a question I need to ask. So which drugs block this enzyme HMG CoA reductase? It's the statins. Okay, it's the statins that block that drug. So make sure you understand cholesterol before you go to take this exam. All right. Okay, so the answer is D. Go to the next one. So here the next one is the active versus facilitated diffusion. Okay. So the question is, also these are the stuff that you need to know. Okay. Active diffusion, facilitated diffusion, and passive diffusion. Okay, there's also passive. So you have to know all those. Okay. And you have to know passive has several forms. You have to know the forms of passive diffusion. So which of the following characterizes both active and facilitated diffusion? Okay. So you have to know both active. So which is in common? So that's what the question is asking. Which of the following, okay, is a characteristic of both active transport and facilitated diffusion? So here you have to know diffusion itself, active versus facilitated. Active means you need energy. When you're active, you spend a lot of energy. So you need energy for active transport. Facilitated diffusion, it means there's already a door, okay, like a carrier. This one has a carrier, but there's no energy. It's like just a door, pass where glucose transporters, like glucose can pass through that channel without needing energy. Okay, so that's active energy. Facilitated meaning you need a carrier, okay, but you don't need energy for that. So because they are all enzymes, okay, these activities, usually if you add something, for example, in facilitated diffusion, you have one molecule trying to pass through a channel, a door. Now the analogy is, if you have so many people trying to fit into the door, okay, at once, then you have competition. All right. So some people go in, some won't, and you have people who are faster than the other ones. Okay. So you have the people who are faster passing faster than the other ones. So there's a competition. Okay. The same with active. Okay. So the answer here, what I'm trying to explain is, the answer is competitive inhibition. Why is A wrong? Because hydrolysis of ATP is energy, remember that, is when you hydrolyze ATP, you are spending energy. So this is active, okay, which is not correct for facilitated diffusion. So this is, A is wrong. C, transport biodirectional. So this is wrong. It's not a two-way street whereby the molecule comes in and then comes out in the same transport. It's not bidirectional, especially for active, okay, for active transport. So transport against a concentration gradient, okay. This is not true. This is not true for active. Active, you can transport molecules against concentration gradient because you are using energy to pump it. So you don't really need a concentration gradient for that. Okay. So make sure you review this. Okay. All right. Let's see. So here the answer is B, as I mentioned. Let's move on to the next topic. So our next topic is well, I gave you the answer already. If you missed that, then we can go back. So membrane transport, okay. This I love this membrane transport, but you have to understand membrane transport, especially when regarding to LDL, okay. Low density lipoprotein particles. Okay, so low density lipoprotein particles get into the cell by what? How do they go in? Do they go in through active transport, facilitated diffusion, simple diffusion, cell to cell fusion, or is it receptor mediated endocytosis? Since I already put the answer there, so the answer here is E. Okay. Receptor mediated endocytosis. So let's break down the question. So receptor mediated endocytosis. So let's start with endocytosis. What does endocytosis mean? Meaning to take in. Into cell. Okay, to take into the cell, but it's mediated by a receptor. So there's a receptor, like a guide, okay, which is going to facilitate the particles to go inside the cell. So now let's see how it works. So you have a cell here, okay, and you have, let's say you have small particles of LDL, okay. So what happens is you're gonna have a signal, okay. So you have one LDL receptor here, okay. Remember we say it's receptor mediated, okay. So this LDL is gonna come and touch here. When it touches here, it's gonna induce other LDL receptors to be expressed on the surface of the cell. So you have now many receptors because now you have so many other LDLs coming in, okay. So now and then the other LDLs, they attach also here. They attach here. Okay. Now they have to go inside the cell. So now you have a protein called clathrin. Okay. Usually it's hard for me to pronounce it, but it's a clathrin. Okay. It's like a claritin almost, but clathrin. Okay. It's like this, clathrin. Remember clathrin, okay? This protein actually goes onto the surface of the cell here. Let's say this is clathrin here, okay? And it makes the cell to have like a small depression. So now it becomes like this, okay? And the clathrin is here, and inside the depression you have your receptors, okay. So the more clathrin you have, you're gonna form like a small vesicle which go inside the cell. Now you have like it goes inside the cell, you have like these receptors inside, okay. So the point I'm trying to make here, even without going into details of membrane transport, you have to know the protein clathrin, okay, that it helps to form vesicles to transport molecules inside the cell, okay, especially like LDL. All right. So make sure you review membrane transport of LDL. Okay, and another one is another one you have to review. Okay, is anomeric carbon. What's an anomeric carbon? Okay, so you have to know what it means. So an anomeric carbon is the center of a hemiketone, functional group. Okay, and I'll explain what it is. So fructose. So you have to know what the fructose molecule looks like. If you don't know it, make sure you at least draw it, and to find a way to memorize both fructose, glucose, and galactose molecules. Okay, the differences are not that much. Okay, especially between the galactose and the fructose. So make sure that you at least find a way to memorize these structures, okay? Because these are like 3 points. So now, if you know the structure, it's fructose is a 5 ring structure. It's like okay. It's 5-sided polygon almost. So you have your carbon, you have your oxygen here, and then you have some carbons here. So some of you are not used to stick figures. Let me draw where you have at least some carbons. Okay. So you have carbon here, you have carbon here, and then you have oxygen here. Okay. Now, and then you have OH here, okay, and on this carbon you have OH, but you also have CH2O. Okay, so anomeric carbon is a carbon which is surrounded by, or which has, okay, both is attached to oxygen and also is attached to OH. So in the middle is going to be a carbon here, okay? So which carbon here is attached to both oxygen and OH? That's an anomeric carbon. So it's this carbon here. So this carbon is the anomeric carbon. Now, how many carbons in this are attached to oxygen and OH? So this one is only attached to OH, so it's not attached to oxygen. This one is only attached to carbon dioxide, but there's no OH. Okay. So fructose has only one anomeric carbon. Okay. So answer here is B. Okay. Now, just to draw it again, you have carbon here, you have another carbon, you have carbon here, and then you have oxygen, then you have OH here. Okay. Here you have OH, and you have CH2O. Okay. So remember, just for clarification, you have this carbon is attached to both oxygen and OH. So that's an anomeric carbon. It's only one. There's no other one. So for you to answer this question, as you can see, you need to know the structure of fructose, glucose, galactose. Okay. Mitochondria, as I mentioned, it's my favorite, and there's always questions on mitochondria. Okay. So the question here is the chemical energy. We have already reviewed the mitochondria in the previous slides. The chemical energy generated by mitochondria electron transport results from which of the following? Excess hydrogen. This is not hydrogen. This is a proton. Okay, because it has a plus. Excess proton in the matrix. Formation of thioesters in the matrix. Hydrogen proton gradient across the inner membrane, or conformation change in the inner membrane. No. So the answer is this one. Remember, I told you when you have let's say this is a mitochondria, you have inner membrane here, you have outer membrane, and then you have this here, the enzyme, the complexes. Okay. So as you transport electrons across, then hydrogen comes, the protons come inside. Okay. And this gradient, okay, is called a chemical gradient because it's, protons. It's called a chemical gradient. The other gradient is electron, so that's going to be electrical gradient. Okay. So here our answer is C. All right. And okay, so that's the end of it. If you have, please, this, I know biochemistry is overwhelming if you haven't taken it, but this gives you like some areas where you need to focus on certain key things that you need to focus on when you go to take the dental boards. Okay, so if you need assistance in terms of guidance or tutoring, let me know. I've been tutoring for a while. Okay, I do work. Okay, so when I make these videos is because I love doing what I do. I love making people succeed. Okay, or helping them succeed. And usually what you need is the correct guidance in this case. So if you have any questions or you need guidance, okay, you can contact me. There's the email in the slides. and then we can set up a schedule of how you can tackle these boards. Okay. I hope you learned something, and I hope it helped. Okay. And good luck.